So hi everyone and welcome for those of you who are here in person. And please let us know on the chat if the audience on Zoom can't hear us. And also big welcome to the audience on Zoom. So I take great pleasure in introducing Dr. Archana Venkataraman as our speaker today. Dr. Venkataraman is a John C. Malone Assistant Professor in the Department of Electrical and Computer Engineering at Hopkins. She directs the Neural Systems Analysis Lab and is a core faculty member of the Malone Center for Engineering in Healthcare. Dr. Venkataraman's research lies at the intersection of AI, network modeling, and clinical neuroscience, and her work has yielded novel insights into various debilitating neurological disorders, such as autism, schizophrenia, and epilepsy. And her long-term goal is on improving patient care and bringing all these fascinating technological advancements to the clinic. Dr. Venkataraman completed her BS, her master's, and a PhD in electrical engineering, all at MIT. And she's throughout her career won several awards, and I'm just listing a few here. So she's a recipient of the MIT Provost Presidential Fellowship, the Siebel Scholarship, the NDSEG, the National Defense Science and Engineering Graduate Fellowship, the NIH Advanced Multimodal Neuroimaging Training Grant, which I'm guessing is a T32, the CHDI grant on network models for Huntington's disease, and the NSF Career Award. So Dr. Venkataraman was also named by MIT Technology Review as 35 innovators to watch under 35 in 2019, which is itself a tremendous accomplishment. So let's welcome Archana. So today she's going to talk to us about deep learning methods for brain connectivity modeling, where the data is coming both from structural and functional MRI. So let's
Thank you. Thank you, Shantanu, for the invitation to speak here, as well as the kind introduction. So it's been really fun being in Los Angeles these last few days. It's the first time I visited for a very, very long time. So it's been quite a pleasure to see the city in person, thankfully. So as Shantanu mentioned, today I'm going to be describing some of the work in my lab, primarily involving connectomics, and especially how do we translate some of these really cool new methods in AI onto these domains for many different applications. So just to set the stage, I can't. So just to set the stage, deep learning has become pervasive as a technology across many fields, from computer vision to speech, natural language processing, imaging, even into material science, genetics, etc. And the rise of deep learning has really been fueled by two complementary things. So on one hand, on one hand, we have really improvements and advancements in computing itself. So this is better hardware, parallel processors, GPUs, the ability to cloud compute, so send your algorithms and models to train remotely, etc. Now, in complement to the sort of hardware computing aspect, one really subtle point and somewhat underappreciated is that the rise of deep learning also depended on sort of a critical mass of data. Right? In kind of the mid-2010s, there was a large effort to curate really open source large data sets, and that's what gave deep learning a foothold as a technology that could span many domains. And in fact, if we focus on this data aspect and we draw a very cartoonish little matrix here, where on the x-axis I have data size and curation. So this could be number of training samples or kind of the level of human annotation and human input that's needed to parse that data. And on the y-axis is complexity of the task that we're trying to solve. What we see is that many of these kind of open source challenge type data sets really populate that lower triangle. So either we have a fairly modest data set like MNIST, but it's also a really straightforward problem, or we have a larger data set to solve something more complex, or in the case of cityscapes, we just have a really, really fine-grained human annotations. Now, I'd argue that many real-world problems that we care about actually live in that upper space, and one of those problems is parsing the human brain. So understanding structure, function, interactions, and particularly differences in health and disease, right? So this is a domain where we have a really complex problem that we as humans don't necessarily know too much about to give adequate training labels. And on the other hand, it's a problem domain in which we're naturally limited in terms of data. And so my lab tackles these challenges across what I call the scientific continuum. So just a wide variety of projects. And these range from foundational, so just trying to parse functionality, build tools for computational neuroscience and multimodal integration, to more translational projects where we actually want to extract some information for a clinician, to exploratory projects that actually move away from neuroimaging, but try and develop a stronger understanding of either the brain body or just cognition itself. So today, this is the outline for my talk. And so today's talk is going to focus on connectomics. So this is functional connectivity, a little bit of structural connectivity. And these three projects that you see here, so these are the three modules of the talk, are going to span a little bit of that continuum. So kind of from that foundational and then looking at deep learning in the context of translational applications as well. So I'll start with what's probably my bread and butter from my PhD training, which is developing models to parse connectomics information, particularly for understanding neuropsychiatric disorders. And here our goal is to kind of move away from case control prediction, but actually characterize some complex clinical phenotype. Now, so we're all on the same page, I'll just describe the data that we're working with briefly and some of the challenges. So this work will rely on resting state fMRI data, or rs-fMRI. So we can think of fMRI data as essentially it's used to measure brain activity over time. So from a sort of a data orientation standpoint, we can think of it as a collection of volumes over time, where each volume has a relatively high spatial resolution between 2 and 5 millimeters cubed and a good field of view, so whole brain coverage. But on the other hand, the sampling rate, so the temporal resolution, is fairly coarse. Now, as I mentioned, this work will rely on resting state fMRI. And so as opposed to a conventional task paradigm, here the subject is lying in the scanner and they're asked to kind of lie quietly while not thinking about anything in particular or moving, etc. So there's really minimal or no external stimulation. And here in this particular data, our feature of interest is really the co-activation or the synchrony or the relationship between the time series in different parts of the brain. And the simplest and most popular metric used to quantify that is a Pearson correlation coefficient, which is the rho ij on the slide. Now, we can aggregate these Pearson correlations over all pairs of regions that we might care about in the brain. And what we obtain is a whole brain connectivity matrix that you see as the matrix on the right. And from that, we can do subsequent neuroscientific and clinical inference. So very early work focused on trying to parse different functional systems in the brain, which you can actually identify quite robustly in resting state. So here I've illustrated 3 well-known functional systems, the motor, the default, and the visual, that were identified because they're really highly correlated with each other or with themselves. And you can think of moving this on to more unsupervised methods, etc. Now, if we look at kind of the clinical characterization in this connectomic space, I would argue that we have fairly blunt analytical tools. And specifically, these methods really fall into 3 categories. So the first category is to really identify statistical group differences. So here we might process the data and extract some features of these correlation matrices. And the analysis strategy is to just compare, is the distribution the same or the different between patients and controls? Another suite of methods looks at just a simple regression against a single phenotype of interest. So based on the problem, it could be sort of response to medication or deficits, etc. Through, again, simple linear regressions. And then finally, with the with the rise of machine learning, there's been an emphasis on patient versus control classification. So here we can take our features and use some sort of classification algorithm to draw a decision boundary or to say, is a new subject a patient or a control? And to a large extent, even with the growth of deep learning, many of the problems really fit into this kind of patient control discrimination or binary discrimination framework. Now, I'd argue that while these methods have certainly been very powerful, very popular, and have yielded many new insights, they don't really capture the complexity of the problem. And specifically, neuropsychiatric disorders like autism actually are fairly complex, and they consist of many different deficits and behavioral symptoms across many domains. And every patient is going to have a unique manifestation. So moving forwards in terms of understanding these psychiatric disorders, it's incumbent on us to figure out how to obtain a more comprehensive and holistic patient characterization. Now, this is going to be our motivation and our goal for this particular project, and we're going to incorporate two other pieces of information. So the first is dynamics. So it's been shown through a variety of studies that the brain transitions through different states over time in the resting state data. And these states are really, the hallmark of these states is that certain networks are more active at certain time points, and then other networks will phase in and out, right? And so we'd like to use this information because it could be that there are certain intervals of the time series that are more relevant for understanding the disorder than others. The second piece of information is structure. And so here, similar to how we have a functional connectivity matrix, while using these Pearson correlations, we can use a complementary modality called diffusion MRI and a procedure known as tractography or fiber tracking to obtain a structural brain graph. So this is anatomical pathways between different regions of the brain. And the field of structural connectomics has done many of the same techniques of statistical comparisons, regression, and classification in the clinical space. And in fact, multimodal approaches, so integrating structure and function, tend to impose an artificial separation between the modalities. So they'll process the function data and structural data separately, and then try to draw post hoc correspondences between the features. Or alternatively, they might concatenate them and try and then feed the sort of concatenated data through a model. Now, I would argue that this, first, it's a little bit ad hoc, but also it doesn't really take into account that the data themselves are telling you different things. So one about direct connections, one about maybe indirect connections. So our strategy is actually to use anatomy as a regularization for the functional networks. And I'll describe how we do that as we move through the talk. So with all of that as a background, I'm gonna present the first model. So this is a model developed by my graduate student Shimona at Johns Hopkins and our collaborator Stuart Mostofsky at the Kennedy Krieger Institute. And as you can see, the model has two parts. So our first, the top component, so the gray box on top, is a representational term, which actually uses classical techniques. And here, the benefit is that we get the structure of a model-based information, model-based prior, and we also get some interpretability in terms of how we extract information. The bottom portion is a predictive term that uses deep learning based on that representation on top in order to predict multiple behavioral deficits simultaneously. So I'm going to build up each block for you, starting with the top. Okay, so the input data to our model for that dictionary learning term is going to be our dynamic correlation matrices. So here, instead of computing a Pearson correlation over the entire time course, we'll chunk up the signal into some small windows, and then we can compute a correlation matrix over each window separately. Now, we assume that these collection of correlation matrices can be represented by contributions from a sparse set of subnetworks B, right? And so the nice thing about this representation is it gives us natural interpretability because each column of B is like a heat map that can be projected back onto the brain. And so that tells you in that particular basis, which regions are highly correlated with each other and anti-correlated with another set of regions. So it's very similar to some decomposition strategies you would see in the literature. Now, of course, this is a dynamic process. And so at every point in time, we'll have a different contribution of these subnetworks. And that's captured by these patient temporal network strengths. And so this will be something unique, estimated for each time point and each patient separately. And then finally, this is kind of an outer product representation. So to complete the representation, we have that basis matrix transpose. So this is the overall framework. It's very similar to common principal components. So it gives you kind of elementary decompositions. And then we're going to build on top of it with our other information. So as I mentioned, here's the functional pipeline, and we want to incorporate structure. And we're going to do this with a regularization. So specifically, we can take our diffusion data, compute our brain structural graph, and use the graph Laplacian as a regularizer on the function. And mathematically, I'll kind of highlight what this does in a few slides. So going back to our model overview, so I filled in the top box, which we call a structurally regularized dynamic dictionary learning. So now let's take a look at that predictive component. So the salient feature for this predictive component are the sort of the variations in the bases contributions. So that matrix C. So the input to our predictive term is going to be the diagonal elements of C, and which you can reshape, extract each separately and reshape into this K by T matrix. So here, K is the number of subnetworks in our particular decomposition, and T is time. So every column is going to be an input feature at that point in time. So we feed this through a recurrent network, and we use LSTM because it's fairly robust. And we get out hidden representations at each point in time. Now, these hidden representations will go through two parallel networks. The first is a predictor network, which is just, in our case, it's based on a simple ANN. So this predictor network will take the hidden representation and for every point in time, give you a phenotypic prediction. So that's the vector Y. So Y can be whatever is reasonable in your problem domain. So this could be just simple case control. It could also be, in what we have of interest, kind of the sort of quantitative metrics across different domains of deficits for the patient. It could be something like treatment response. So it's actually a very flexible framework. And then parallel to the predictor, there's an attention network. And so the attention network tells you how much to weight that particular time point. So if a particular interval is very informative, it will have a high attention weight. And if it's not informative, it'll have a low attention weight. And our final output is just a weighted average across all of the element-wise predictions. So here is our full framework. This appeared in MICAI in 2020, and there's a recent neuroimage journal extension that just came out if you're interested. Now, if we peel back the hood a little bit and try and understand what's going on, so our main objective function has two terms. The first term is that structurally regularized dynamic dictionary learning. So that's a reconstruction error. And if you notice, that reconstruction term is not a Frobenius norm, it's weighted by the graph Laplacian. So effectively what's happening is we're going from an isotropic loss. So a Frobenius norm we can think of as isotropic. So an error in every direction is equally weighted because you're taking a sum. And with the reweighting through the graph Laplacian, effectively it sort of compresses that loss shape and it emphasizes directions or entries along which there's an anatomical pathway. And it de-emphasizes more indirect connections. And so that basically is our way of saying, well, the sort of anatomical pathways, that's the infrastructure for the connections or for the signaling. So we trust those a little bit more than the indirect connections, which could be due to a variety of effects. Now, the second term of our loss function is the deep neural network. So that's the prediction loss. In our case, it's mean squared error loss. It could be, if you were in a classification setting, like a binary cross entropy. And then finally, there's two regularizers for this problem. The first is a non-negativity constraint on C, and that gives us a well-posed solution because our inputs are positive semi-definite. So we estimate a positive semi-definite representation. And the second is an orthogonality on the bases. So this is similar to a PCA where we want the bases to capture maximal variance in this higher level sense. And so we have that orthogonality. Now the unknowns in this representation are the bases B, the coefficient C, and then the neural network weights theta. And we optimize all of them jointly using an alternating minimization strategy. So here we update B using, the update for B actually is nice, it is closed form given the other parameters, and it's a Procrustes-like solution. The update for C is actually what couples the two terms, this generative classical model and this deep neural network. So effectively it's a gradient-based algorithm and a gradient-based update. And one term of the gradient will come from the dictionary learning, one term will come from the neural network. And it turns out we can use Atom just to fuse those two gradient terms and automatically update C. And then finally, we update the neural network weights just using whatever standard algorithm you favor. So we use Atom, but you could use SGD as well. So the data set that we've used in this paper and in this talk for evaluation is neuroimaging data from 58 ASD autistic patients, where we have resting state fMRI and diffusion. The features that we're trying to predict, so that clinical phenotype, will consist of 3 diagnostic measures for ASD. So the first is ADOS, which is the very standard behavioral exam used for diagnosis, administered by a trained psychologist. The second is SRS, which is a parent report or a parent questionnaire that they'll fill out based on the child's behavior. It's scored. And it's targeted more towards social functioning and social reciprocity. And then the final praxis is a motor exam. So it measures things like visuomotor, hand-eye coordination, gestures, imitations. And this was a test developed by our collaborators who are very interested in motor deficits in autism, which is a defining feature, even though it isn't a standard clinical diagnostic feature. So with those 3 as our phenotype, our evaluation strategy is 5-fold cross-validation. And we compare with a couple of baselines in this talk. So the first is the BrainNet CNN architecture. So this is an end-to-end deep learning model where effectively instead of a standard convolution, which is a little grid, kind of a little square kernel that moves around in a grid, here the convolutions are cross-shaped filters. And then we fuse structure and function to get the output. And the second is data-driven feature extraction. So here we kind of fuse structure and function in some sort of joint PCA, and then feed those representations into the LSTM-based predictive model from earlier. So I'm going to show the results as a series of scatter plots. So the first scatter plot is for the BrainNet CNN in this case. So here, we have the 3 scores as 3 different columns, ADOS, SRS, and praxis. The red points are the training data, and the blue points are the testing, sort of the testing predictions. And what you can see is it does okay-ish on ADOS, but then the generalization is actually quite poor on the other 2 metrics. So here, measured as x-axis, predicted as y. So ideal performance is the y equals x, so the diagonal dashed line. And then the 2 flat lines are just the population means of the 2 scores. So we observe a very similar trend for the PCA and LSTM. So here, actually the performance on ADOS is quite good, but then the generalization on the other 2 scores is completely flat. It's just predicting the mean value. And we have empirically have observed this with the many baselines that we've tried, that we can tune the parameters of the baselines to do well on one of the scores, so one of the domains, but then it'll fail on the other 2. So kind of this joint prediction is actually quite challenging. Now here are the results of our method, and it's interesting. So we're able to at least capture data variation and the rough trend in each of the scores simultaneously. And that's reflected in the quantitative metrics, which are median absolute error and normalized mutual information, which are almost uniformly higher than all of the baselines. Again, with the exception of doing really well on ADOS for the PCA LSTM. Now, one nice part of our model is that the subnetwork bases that we're estimating, again, can be viewed on the brain and interpreted. So we just took a look at 4 of the subnetworks that we recovered. And what we note is that they correspond to very well-known systems and sort of interactions between well-known systems. And many of these networks have been reported in the autism literature as well. Now, finally, we can also take a look at our dynamic information. So what on the left, we have the attention weights over time. So patients are vertically stacked and then time is horizontal. And once again, we note that for every patient, it's kind of a different interval of the time series, which is selected as relevant. And so that supports the hypothesis that that dynamic evolution might be very valuable when we're parsing some of these complex disorders. And then on the right, we have, for a single patient, I've just plotted those coefficients C over time. And we also note some variability in how those coefficients track. So again, future iterations, we can take a look at really carefully parsing which networks are implicated across patients and the dynamics of that. So I think moving forward with this project, what we're really interested in is applying it to a more diverse cohort, potentially with many disorders, and seeing are there overlaps in the representations we learn across different neuropsychiatric disorders, and then can that be used to provide a more holistic characterization of sort of mental health deficits in general. So moving on. That first project, again, it was much more developing tools and techniques that can be used to kind of explore different computational neuroscience problems. The next 2 projects will actually focus on deep learning itself, so end-to-end modeling for 2 translational applications. So these are all going to be based on graph evolutions as well. So the first is deals with pre-surgical planning. So our motivation here is we want to provide information to neurosurgeons about how to safely resect a target, right? So if a neurosurgeon is going to remove, let's say, the brain tumor that you see here, there are a couple of areas that they want to really avoid damaging. And the most common are the motor areas of the brain and the language areas of the brain, right? So if that's cut, often the patient can have very, very severe deficits postoperatively. Now, the gold standard is based on electrocortical stimulation. So this is a procedure where during surgery, the patient is actually awake and responsive, and they use stimulation to try and understand where these targets are. So this is considered the gold standard, but it does increase the duration of surgery. It's fairly taxing on patients, and it's really limited in field of view. You can only probe right where you are operating, right, rather than anything broader. So as a compensation, task fMRI has started to become used as a preoperative mapping tool non-invasively. So here, a patient might be given a motor or language task to do in the scanner, and then through a GLM analysis, the activations associated with that task are provided as kind of proxies for these language and motor areas to the neurosurgeon. Now, the issue with the task fMRI is that many of these patients are actually fairly impaired. And so if a patient cannot perform a task in the scanner, then the resulting activation maps are completely unreliable, right? And so our goal is to use resting state fMRI effectively as a replacement to task. So to try and identify these eloquent cortex regions using resting state. And the reason it's a popular choice, or it's a good choice in this case, is that it's a passive acquisition. So the patient doesn't need to perform anything in the scanner. They don't need to cooperate. There's no successful paradigm, et cetera. It's just quiet acquisition. Now, there's, I think, 2 categories of work that have been used for resting state mapping of eloquent cortex. The first is based on group ICA. So effectively, the fMRI data for across patients is concatenated, group ICAs run, and then the resulting spatial maps are sort of used to determine where these areas lie. Now, the issue with group ICAs, it actually requires a lot of expert intervention. So the group ICA, the spatial maps need thresholding, and someone needs to select among 50 components, which is the component of interest. And so that will fall to a radiologist, and there might or might not be success for a given patient. The second method is actually based on deep learning. It's based on a multi-layer perceptron. So here, resting state fMRI data is transformed into several seed-based correlation maps, which are used as input to a multi-layer perceptron to kind of predict where do the regions fall among well-known systems, so attention, visual, motor, etc. Now, the main drawbacks to this approach is, once again, there's some expert intervention, so we need to have good seed definitions. And it also doesn't really take into account network associations because it's just a fully connected layer. And partly due to these limitations, there's been really minimal quantification of sensitivity and specificity of the methods. It's been very heuristic in terms of publication. So it's really unclear how well they perform under duress or in a real world setting. So our approach, as I mentioned, is going to rely on deep neural networks. And so this is work developed by my graduate student, Naresh, at Johns Hopkins, and our collaborator, rs-FMRI, who is a neuroradiologist at Johns Hopkins. So as you can see, the model itself has three components. There's an input encoding, a classification branch, and then a dynamic component. So I'll start with the input encoding based on the convolutional networks. So the input to our CNN is going to be dynamic connectivity matrices. So here we can parse the brain and extract average voxel-Wells time series in every region. And once again, we're going to use sliding windows to obtain these input matrices. So from 1 to T, we chunk up the time series and obtain a correlation matrix at each one. In this case, in this particular work, we do exponentiate just so all of the matrices, the input values are positive rather than negative and positive. Now, unlike the first project, one defining feature of this patient cohort is the tumor. Right? And many of these tumors are large and they encroach into gray matter. And since it's really unclear what a fMRI signal in a tumor means, we don't really want to use it too much in our analysis. And so we'd use a very, very simple strategy. We just zero out the rows and columns for every patient that correspond to the tumor. So these are the black lines that are shown on the slide. Now, with that sequence of zeroed out connectivity matrices, we're going to feed each one into very similar to the BrainNet CNN architecture. So the first layer is an edge-to-edge layer. So these are the cross-shaped filters. And this cross-shaped filter, in some sense, it has the ability to capture network theoretic properties. So if you compute a network theoretic measure like a degree, it actually can be implemented through these cross shapes. And so the idea is that sort of in a data-driven fashion, we're learning these kind of weighted network statistics. Now from there, the data is collapsed along one dimension, and we obtain an n by f representation for each correlation matrix over time. And this representation is input into the classification module to try and detect these eloquent systems, and the attention module to parse dynamics. So going back to the overview, I'll now go through the dynamic component. So we have these feature representations. So it's across regions and across time. We collapse across regions to obtain a low dimensional representation across time, reshape that. And once again, we can use a recurrent architecture, your favorite architecture. In our case, we found an LSTM to be robust. And from there, our outputs are essentially two attention vectors, one for language and one for motor, because in our data set, we have language and motor paradigms. And so that tells us, each vector tells us, so at every point in time, how much is this relevant to language versus how much is this relevant to motor. And if you had additional tasks that you cared about in this preoperative mapping stage, we could just add those on as extra outputs as well. And then finally, the features and these attention weights will go into our final multitask classification block. So going back to the final overview, we can now focus on the Eloquent Cortex detection. So here we have our CNN that generates these features. We just do some dimensionality reduction and get through a very simple ANL, and we have 4 branches corresponding to the 4 tasks that we are able to predict in our data set. And the representation is kind of n by 3 by t. So for every region over time, for every class. And effectively, we kind of multiply that using a Hadamard product with the attention. So here, effectively, we're going to select for every branch which elements of the time series are relevant. And then our final output are these spatial maps, which you see on the right-hand side of the slide. And so what it does is it classifies every region into one of 3 groups. So there's either the tumor, which is just recovering the rows and columns of 0. So it just helps the model. It tells the model just recover this and allows the rest of the network to focus on discriminating the eloquent cortex. The second is the eloquent class for each branch. So the language in one case, and then the 3 motor tasks separately in the other case. And then finally, just the background gray matter as the 3rd class. Now, this model is very flexible. So again, if you had more tasks that you cared about to map, so maybe visual systems or attention networks, you could just add more branches to the model, and it would be able to infer a new map for that particular class. It's also really nice because based on how the loss function is, which is a concatenate, where is kind of the sum of all of these losses, we can actually train this model in the case of missing data. So if a particular patient that we have during training was not given a task because it was deemed not clinically relevant, we could still update the rest of the network based on that patient, and therefore kind of maximize the amount of information that we're using from our data set. So here is the kind of final model, and this appeared in the MLCN workshop in 2020. And there's been kind of a journal version of this that just came out, as well as an extension that was in IPME using spatial attention as well. So our evaluation is on 56 patients from Johns Hopkins Hospital that have tumors, and they underwent preoperative mapping. So we had T1 MRI, which was used for tumor segmentation through a radiology fellow did this for us, and they were confirmed by the expert radiologist. And then we have resting state fMRI. Now, in terms of model training, we need some notion of ground truth labels. And so that was derived from task fMRI for these patients. All of our activation maps were confirmed by the radiologist. And so there are 4 tasks: language, and then 3 motor tasks: finger, tongue, and foot. Notice that we have, so in brackets is the number of patients with each modality, so each task. And so notice that it's not a complete data set. So definitely some patients are missing some of the motor tasks, and some patients are not. Also very interestingly, so our language patients there are 2 cases. There's cases where the language is only on the left hemisphere, which is the most common in the general population. Most people have left hemisphere dominant language. And then there are a few patients with bilateral language networks as well. And finally, we use the Cortex atlas as our region parcellation. We have played around with different atlases, and for the most part, it doesn't change the overall performance. And finally, we have 3 baselines that kind of pull apart one aspect of the model. So our first baseline is is purely classical. We use a PCA for dimensionality reduction, and then an SVM for classification, multitask classification. Our second baseline omits the dynamic component, but it has a very similar architecture just for static connectivity. And then the third baseline omits these edge and node-based convolutional filters, and instead uses an ANN with the same number of parameters as the encoding block. So here are the results from 8-fold class validation. And because this is quite a bit of numbers to parse at once, here's some salient information. So overall, our model does almost uniformly better than all of the baselines across all of the tasks that we're aiming to classify. And it does particularly well in language. So this actually is because what we saw is that kind of the idea of networks phasing in and out was quite important. And so that helped us localize the language network much more accurately. Another interesting experiment we did is we trained the model only on the left hemisphere patients, and then we tested them on the bilateral patients. And what we found is, to some extent, the network is able to automatically recover the bilateral information, despite never having it in the training data as well. Oh, and this is just, so here the blue is the ground truth based on the task, and then the yellow is what's predicted for the model. So we're able to recover that bilateral activity. And finally, we can look at the network attention weights, and what we note is that, and it's kind of highlighted in the blue box, that motor and language are phasing in and out of each other. So segments of the time series where motor is most relevant, language is kind of de-emphasized in terms of our prediction, and vice versa. So this dynamic evolution is also very, very helpful for determining these specific systems in the brain as well. And then moving forward, we actually have, we've developed extensions of this work. And right now we have some funding to look at this in a more prospective fashion. So we're going to be scanning brain tumor patients before and after, or before surgery, running these models. And we're also doing neurocognitive testing to see if the predictions of our model are correlated with postoperative deficits in language. And then finally, this last few minutes, I'll describe very, very recent work that we've done, also in translation and taking this idea of localization. And here the goal is to determine seizure onset in epilepsy patients. So again, as stated, this clinical application deals with epilepsy. So epilepsy is one of the most common neurological disorders, and it's associated with really devastating quality of life and mortality rates. Right? Now, most patients are actually treated with medication, but about 20 to 40% of them are considered medically refractory, so they don't respond to a variety of drug therapies. And so here, the most common subsequent therapy is to see if we can identify a single origin for these seizures, then the therapy is actually to surgically remove it. And that's been shown to have pretty good rates of seizure freedom. Right. And so epilepsy is really a long clinical pathway. And what's pretty salient is that the result of that is only a 50% success rate. And so the idea for this project is, can we use other types of information to get a better idea or a better localization of the seizure onset to give to clinicians to move in that pathway itself? And the working hypothesis of this project is that if we have a patient with epilepsy and they're having seizures and it's chronic, it's long-term, it should impact brain connectivity in some manner. And so that's essentially what we're going to do. We're going to use connectivity information to try and detect these regions. So this is sort of follow-up work that Naresh did using a slightly different architecture. And our collaborators here are David Shu and Raheel Ahmed from the University of Wisconsin in Madison. So here, because we have a very small data set and we're really looking at kind of a very severe deficit, we just focus on static connectivity, but we do use anatomy. So the static connectivity matrix, so correlation matrix, we feed it through a couple of graph convolutional layers where the graph convolutions are guided by the anatomy, which we can estimate from diffusion MRI. So here, the layer outputs, it's basically these graph convolution or graph neural networks are very similar to an artificial neural network, but there's a pre-multiplication or a guide with the anatomy. And then the output of those graph convolutions are fed through a fully connected layer. And there's two branches. The first is our prediction of onset. And the second is a subject-specific bias, and they're kind of fed together. And for every region, we have a prediction: is it the seizure onset or SSE, or is it not the seizure onset? Right? And that turns out that bias is actually quite relevant. So effectively what it does is, because we're feeding it through a sigmoid, it tunes the threshold of the sigmoid in a data-driven fashion. So it tells us where to draw the threshold of detecting seizure onset versus not. Now, beyond the architecture, if we look at the loss function, there's really two components. There's a weighted cross-entropy loss. So that is just for every region, are we getting the seizure onset correct or not? And there's also an asymmetry loss. So basically, the idea is that these seizure onsets, if we believe it's focal, it should occur in one place in the brain, and we should not see it, that same prediction, on the contralateral side. So they should be lateralized predictions. And finally, there's a couple of hyperparameters, so the class weighting and the asymmetry, which we actually tune on the Human Connectome Project dataset. So we take that dataset and we insert artificial seizure onsets, run the model, and then use that to set the hyperparameters. So here we have a very small data set, 14 pediatric epilepsy patients from the University of Wisconsin. We have preoperative and postoperative T1, and we use the surgical resection area as ground truth, effectively. That's kind of the best ground truth we have at this point. And they also have resting state data. Now, the anatomical template we actually construct from the HCP data set. So we use 50 randomly selected patients from HCP, and we use that as the anatomical template throughout the neural network. And we use Brain Tome Atlas, which is a reasonably fine atlas for our parcellation. Oh. And then one really salient feature is that our 14 patients are extremely heterogeneous. So on this table, I just have listed the expert determined seizure onset for these patients, and they're all over the brain. So every patient kind of has a different onset across frontal lobe, temporal lobe, right hemisphere, left hemisphere. And this actually differs quite a bit from the previous project, where we'd expect motor and language to be roughly in the same spot per patient. And here that is not true with respect to the seizure onsets. So here are just some very preliminary experimental results that we've gotten from this work. And once again, just to highlight a few key points, if we run this full model, we actually get really decent sensitivity in AUC. And the reason sensitivity is a little low is because we're using seizure resection as our ground truth. And in epilepsy, the resections are quite large. They resect far more than the actual onset themselves. And so this kind of has a dampening effect on some of the sensitivity measures. With that said, compared to ablated versions of the model, where we either have no asymmetry, no subject-specific bias, and no graph information, we do quite well. So the combination of all of these methods and information together really helps us achieve something that's on the boundary of actually being clinically relevant and useful. And just to look at a couple of examples, so here, red is ground truth, blue is what's predicted by the network. These are testing patients using Leave-2 cross-validation. And we can see that the network actually does a very reasonable job of trying to finding these seizure onset locations. And that's fairly consistent across these patients. And so again, with further testing and exploration, this could be a modality that's really easy to incorporate in a clinical pathway and could be helpful in the context of epilepsy. So before concluding, I'd like to thank my fantastic lab, my collaborators, and our funding agencies. All of the folks underlined actually contributed to this presentation and the work presented. And finally, a plug. So if you are using EEG data, I would highly recommend you check out our visualizer, which was developed by my undergraduate student, Danielle, at Johns Hopkins. So this visualizer allows you to load and view EEG signals. It allows you to load deep learning models, so predictive models, run them on the EEG, and then plot the resulting outputs, both on the time series and on the scalp. So you can save all of this and use them in presentations and papers as well, which we've done. So it's freely available for download on my lab website. So to wrap everything up, I've kind of highlighted 3 different projects that span this spectrum from developing computational tools and models that are generally applicable to very concrete neurosurgical problems. And so what I'm hoping is that in the future, we as a field start thinking about what do we do with fMRI data now that we have these tools to analyze it? What types of problems can we solve? Can we actually make an impact on patient care and really move the field forward in that respect? So with that, I thank you for your attention, and I'm happy to take questions.
Yeah, I'm hoping you can elaborate a little bit on what you meant by ground truth.
Oh, for epilepsy?
Yes.
Okay, so for epilepsy, we're using.
Sorry, do you mind repeating the question so for the Zoom audience? Okay.
The question was to elaborate a little bit more on the ground truth that we used in the epilepsy project. So the two bottom projects are supervised learning type of approaches, where we have some training data, where we have a ground truth label, and we're kind of learning the characteristics of that, and then applying it on testing data, just given the neuroimaging. So in the case of epilepsy, ideally what we'd want is a ground truth of the seizure onset location. That's actually a very difficult and very precise characterization, which we essentially don't have. And if you look at the clinical workflow, they're really looking at kind of narrowing it down to lobes or certain areas. And so what we did here is we have pre and post T1 MRI, so preoperative and postoperative. And so the area that they remove, it corresponds to where the experts and the clinicians think the seizure onset is. And since we don't really have a way of narrowing it down beyond that area, we just use the entire resection volume as the ground truth.
So you mentioned that this procedure is about 50% effective.
Yes.
If the procedure was effective, does that mean you believe the ground truth?
So the reasons it fails is very complicated. Unfortunately, we don't have postoperative seizure freedom in this case, but that if we did was something we might go back to. So the issue is not always that they took out the wrong area. Sometimes they just missed a secondary foci somewhere else on the brain. So that could be one example. Other times there are regions which don't even look problematic, which over time develop as problematic. So there are, again, many reasons that the procedure could fail, but I think the multifocal aspect is the thing we're trying to target in terms of reducing that risk. Yeah.
In your earlier models, you were showing the kind of this PCA version trying to estimate gamma.
Mm-hmm.
I was wondering if you could explain where in the model you estimate BNC.
Oh, okay. So it's in the alternating minimization strategy. So the question was a little bit more details about where we estimate all of the unknowns in the first project. So this is the objective function. So it's just an optimization function or an optimization objective. And so the yellow that's highlighted are all of the unknowns. So the B is unknown, a priori, all of the C's are unknown, and then the neural network weights are unknown. And so in alternating minimization, the strategy is you fix two and then update the other one. And so we use a fairly intelligent initialization. So we just run like common principal components and use that as an initialization, randomly initialize the neural network weights. And then once we have an initialization, we can fix the values for C and theta, and we update B via a closed form expression. And the actual expression's in the paper if you're interested in that. It's per custis because that weighted loss. There's kind of a Krusties aspect that uses that graph Laplacian. Once we've updated B, then we can move on to updating C, and it's a gradient-based algorithm. So we have one component of the gradient from the first term, one component from the second term, and it's just the sum of the two because they're summed here. And Adam actually naturally will update a parameter if you give it a gradient. So we just give Adam the gradient and it updates that. And then we update the neural network using backpropagation.
Sorry, one more question.
Yes.
What type of machine learning API are you using?
I think this was all in PyTorch.
Archana, since you're on that slide, CTN is a function of time,
Yes,
and you diagonalize it, so it's still, so it's a matrix.
it's a diagonal matrix.
Diagonal matrix, and n here is the number of nodes.
No, it's the nth patient.
Nth patient. So
Yes.
n is actually the patient,
Yes,
and okay, it's a time series, and so you discretize T. into
Yes.
some things.
so in this case, T is the window in the sliding
so
window.
T is actually the window.
Yes,
The window size.
not size, the window index.
The window index.
Yes,
Okay. So the actual so if window index means that the actual temporal fragment.
Mhm.
Where is that? So that's actually CTN.
no, the temporal fragment is in gamma. So the fragment is
gamma?
used to compute a gamma.
Okay.
Yes.
Okay, that helps. So that means when you say you update B via closed form processes, you're solving another problem here for the first term to estimate B, or is B getting estimated in the optimization of this?
B is getting optimized to minimize this loss function.
Okay.
And it turns out when you take all of the gradients
Yes.
and rearrange terms, the update itself is closed form. Yes. Yes.
So we have a few questions in the chat.
Okay.
So if you don't mind going to the chat.
Okay, I can do that.
And then we probably have a few questions at the top. So
Okay.
Oh, yeah.
Recording, everyone. The nodes in the graph neural network. So all of the models are ROI based. So an input node is the region, roughly, whatever the parcellation you hope to use is.
And there was one final question from Shima Mohammadi. Is the brain map actually successful in treating epilepsy, or it's still being tested?
Oh, it's still being tested. This is preliminary work. It's actually one of the first demonstrations of resting state being useful. It will take a lot of testing before a clinician would be willing to use this information.
Okay, so there are no other questions from the audience. Let's thank the speaker again. And